The brain's navigational place in grid cell system. The 2014 Nobel Prize in Physiology or Medicine is awarded to Dr. John M. O'Keefe, Dr. May-Britt Moser and Dr. Edvard I. Moser for their discoveries of nerve cells in the brain that enable a sense of place and navigation. These discoveries are groundbreaking and provide insights into how mental functions are represented in the brain and how the brain can compute complex cognitive functions and behavior. An internal map of the environment and a sense of place are needed for recognizing and remembering our environment and for navigation. This navigational ability, which requires integration of multimodal sensory information, movement execution and memory capacities, is one of the most complex of brain functions. The work of the 2014 laureates has radically altered our understanding of these functions. John O'Keefe discovered place cells in the hippocampus that signal position and provide the brain with spatial memory capacity. May-Britt Moser and Edvard I. Moser discovered in the medial interhinal cortex, a region of the brain next to hippocampus, grid cells that provide the brain with an internal coordinate system essential for navigation. Together, the hippocampal place cells and the interhinal grid cells form interconnected nerve cell networks that are critical for the computation of spatial maps and navigational tasks. The work by John O'Keefe, May-Britt Moser and Edvard Moser has dramatically changed our understanding of how fundamental cognitive functions are performed by neural circuits in the brain and shed new light onto how spatial memory might be created. Present neurobiological concepts regarding superior cognitive functions are based on synaptic neurotransmission and neuronal plasticity. However, the diversity and complexity of neurocortical connections, circuits, maps and their relationships with memory, learning and other superior cognitive functions are not fully explained by the present neurobiological paradigms. Recent discoveries concerning the ability of neuronal cells to perceive and process very weak electromagnetic information suggest a possible role of biophotons generated as a consequence of neuronal and astroglial metabolisms in a wide diversity of cognitive representations. Moreover, the finding that the human brain has magnetite nanoparticles opens new possibilities about the role of these nanocrystals in information processing and memory. In the present chapter, a previously advanced model, neuron, astroglial communication in short-term memory, bio-electric, bio-magnetic and bio-photonic signals, is developed. Based in the ability of neurons and astrocytes to generate biophotons as a consequence of their metabolic activities, I propose the generation of innumerable biophonic-mediated nanoholograms, which are produced and modulated by magnetite nanocrystals associated with neuronal and astroglial membranes in the cerebral neocortex. Specifically, it is suggested that biophotons generated by neuronal and astroglial cells may produce multi-channel holographic images through their interaction with single-domain and/or superparamagnetic magnetite